For that, we have to collect the soil samples. Soil is a really a rich source of bacteria, fungi, and soil is a source for everything. Correct. Everything comes one of the part of, if you see our Panch Tattva, then we are also made up of soil. So part of it. So the soil is a really a very useful source of getting a new culture. Now you can say that everybody has tried from the soil, but the soil flora and fauna, which is right now what we understand and what we know, is just a tip of iceberg. Really, we don't even know 1% of the bacterial flora, when I mean microbial flora of soil. And that is why our chances whenever we want to go for a new product, and for that, when we want to isolate an bacterial or fungal, a microbial culture, what we will do is we will collect a soil. Now, soil as argument can be right that soil, everybody is collecting soil. So what? So what we try to do here is we try to collect it from a various different sources, and that sources can be extreme environments. Yeah. Can anybody tell me what are the extreme environments? In fact, if you are seeing that, you can see the example. Can anybody tell me what is an extreme environment?
Sir, extreme uh, environments with high extreme conditions like where thermoacidophils, where they live very
Ah, good.
high temperatures
Yeah, I think
and
precisely. So,
salinity.
so perfectly, exactly. This is the exact answer. So I take it for granted that all of you are on the page and we are going fine and you all are understanding. So in extreme environments, rightly said so. So, let's take when I actually, I have worked for my beginning career, as I told you at Huxt was this only, and we used to go for collecting the soil. And that's why I'm not telling something which is theoretical. Actually, I have done it. So, let's take the first extreme environment. We had a team and from CCMB, that is in Hyderabad, there is a very good institute. They were going, there is Dr. Shivaji, he was going to Antarctica for collecting the soils, and we have taken soils from him. Now you see, the Antarctica is like, it's a tip, there will all the time very cold weather, right? So they will be collecting the soils from there. And when you bring this soil to your laboratory and try to cultivate the organism, your chances of getting a different kind of a novel organism increases. And by getting a novel organism, our ultimate goal is to get a novel product. So that's one environment. The second environment I can immediately remember is, volcano. In a volcano soils, you will get a very rare organisms, and when they produce the product, I think they will have a multiple advantages. Just these two examples when I'm telling you, and then let me give all the first soil collection examples, and then I'll come back. Then, deep sea, we have taken it from, I think, Goa deep sea. There is an institute of oceanary there, and they have brought us soil samples from deep sea. So the sea samples, sea soil samples were very productive, and you can have this. So, there are two things happens in a sea. There is a sea sample when you take there is a lot of pressure and then halophils. So all these three extreme environments, and yes, most productive or my first patent came from an desert soil. The Rajasthan desert, we have collected the soil, and another is because deserts have another very different kind of high temperature. Volcano is different, and because the volcano soil is having a very different kind of composition as compared to the desert soil. So desert soil will have a very high temperature and there is a selective pressure, and that is why certain type of organisms can only survive. So these are some of the four different extreme environments you select for collecting the soil. Now let me just give you if example why what will be the advantage of your product should be in your mind. If you're looking for a very thermostable product, something which will be very stable in a high temperatures, there is good chance that when you have these thermophils, with an extreme desert environment or from volcano soils, when you isolate an organism, there is going to be a very good chance that they will be producing a product. It's a chance I'm telling, but there is a good chance that it will be producing a novel product which will be very stable at very high temperature. Similarly, now you can use the analogy for sea, which is halophils, that is a very salt tolerant bacteria will be able to isolate using the this soils from sea. So now any questions here? I think we'll move to the after you have collected the soils and you have gone all over the world. Doesn't it appeal you that you have to go to a desert and you'll be going to Antarctica and you'll be going to Goa and then going within the divers to take the soil? It's a very nice challenging job, but really entertaining job. So anyway, questions here. This is the first step of soil collection in a methodology of bioprospecting.
So I have a question.
Sure.
So you just said that, based on certain properties that, we want the product to have, for example, thermostability, you can select the microbe, right? But then even in, let's say, a volcanic environment, there'll be different thermophiles, right? So how would we know which one would work best?
Yes, I think that's a good question. See, in a bioprospecting, when we are collecting this soil, we are just taking a chance, Just let me give you some analogy and then I'll come back to the question. Like, wearing this mask, can it really stop COVID? I don't think so. But yes, I'm reducing my chance. Correct. Taking a vaccine of existing viruses, do you think down the line, few years, we'll be able to, the virus changing every day its protein, PDB binding domain, that is protein binding domain of COVID domain. Is it going to really completely protect? No, but what we are doing is increasing our chance. So in a science, what we do is, probability increase, which now bringing you to a little scientific aspect is probability increase. So when we try to use these soils, we are just increasing our probability. We are not sure about anything. That's how it works.
Okay, so thank you.
Okay. So, the next step is after bringing these soil samples into your laboratory. Now you have come back into from desert of 50-degree desert to and zero or sub-zeros of Antarctica. You have brought the soils into your laboratories. Now that's in 27 degrees centigrade. So the next step, we are now going to isolate the organism. So the next step is treatment of soil. You need to treat the soil. And here there are part A is treating the soil, storing and treating the soil, and the second part will be using this soil for a cultivation. And for that cultivation, you all know the media is. Have you all prepared any media? Nutrient agar, Mekongis agar. Have you at least seen in a petri plate?
Yes, sir. Yes.
Yes.
Yes, sir.
Fine. So, the next step in the step number 2, now we are back into the lab and we are going to isolate the organism. So now I'll give you all 4 examples how we treat that soil. See, if I brought a soil sample from Antarctica, yeah, what I would first thing is storage, correct? All the soil sample around, we collect 100 soil samples from say it's around 70 to 100 square kilometers. So 1 square kilometer once soil sample is collected. So those soil sample will be storing it at sub-zero temperatures. Your minus 40 or minus 70 deep freezer will be storing. And while the soil samples which I have brought it from high temperature or arid region, we will be storing them at a high temperature. So first thing is storage. Yeah. The second point is that while bringing the soils to the laboratory and then trying to work in a laboratory, there is a chance that you have contaminated with the very common organism. And that's very obvious. Like, we are not all the time collecting the soils or bringing the soils and opening in a laboratory under the laminar airflows. We don't do that. So we'll be treating the soils to eliminate and avoid the unwanted bacteria, that is E. coli and many other bacteria and other spores which are just in the air. Aerial flora we don't want, because we have done all this effort to collect the soil sample from a unique place. And that is why the next step is to treatment of soil. And this treatment of soil, there are many 3 or 4 techniques which are available for treatment of soil. But today I will just tell you a very general treatment, and then next class I'll use the slides. In fact, in this PDF, if you go down, you will see different treatment methodologies. But what my first methodology would be to eliminate the organism is, if it is an arid, let me take just example of one, and then you can use that analogy, and during your exam you can expand it if it is 20-mark question onto all the 4 type of soils. But you will be treating say arid and high temperature soil, then I'll keep it at 180 degrees centigrade or 120 degrees centigrade to kill all the other bacteria. But I don't want to kill my forest bacteria, so I'll not be keeping only at 120. I'll be also keeping it at an around 40 to 50 degrees, which was the actual temperature in the desert. And now using these 2 different treatment samples, I will put them onto a media. Now I'll be trying to grow the bacteria, correct? Grow the organism. Then third step. Third step is first collection of soil, treatment of soil, and the most important now, because we need a microbial culture for production and check them whether they are producing a novel kind of compound of our interest. So the next step is will be designing a culture media. Here you have to design your culture media. See, you can take a nutrient agar. Yeah, but then it is going to be again going to give you a large number of organisms, but it may not be very selective. So we will take nutrient agar definitely, where all the kind of organism grow. And then second, we also want nutrient agar is more favorable for a bacterial culture. So for a fungal culture, which are also very important from an bioprospecting point of view, we will use Sebrod Dextrose agar. You all have heard about Sebrod Dextrose agar? See, this detail may not be in your notes, but I think it is good for your knowledge. Sebrod Dextrose agar is an agar with an, sorry, hello, Ardish.
Yes, sir, I was telling that we haven't learned about it yet.
Fine. So Sebrod Dextrose agar, as the name itself, or potato dextrose agar. Probably you have heard of potato dextrose, but anyway, I'll just tell you what it does. What it means is a very high sugar. Yeah, if you know about yeast and fungi, then you know that they can tolerate a very high sugar concentration, and they really need sugar concentration, very high sugar concentration for growing. And that's why we use a 4% sugar dextrose and potato extract. We actually cut the potato, and you can actually make in a lab when you come. You just bring a potatoes, cut it, boil it, and put 4% sugar and an agar in it. So that is how we design different kind of media to avoid the organism. So once these soil samples which are brought from an extreme environment, treated in the laboratory in a very normal way, are further used for cultivation. And just today, I think we can at least touch upon one of the methodologies for See, can you all see this two on a right hand side? You are seeing and some tubes, and then there are some wires kind of thing. They are also actually tubes. Can you see your screen?
Yes, sir.
Yes, sir.
Yes, sir.
Can you just read it, figure 2? What does it mean? Like, just can you read it out so then I'll be able to.
Sir, it just says cultivation techniques. So A is a diffusion chamber, B is a hollow fiber membrane chamber, and C is an encapsulated gel micro droplet.
Correct. Yeah. So I think I'll explain you this diagram. See, this in exam, I think this diagram is very important. So collection of soil sample and all that, I think you'll be easily remember. But these are the two techniques which are here. And on your right hand side, you will be able to see one chamber on B diagram B. You just see there is a chamber with some tubes put it on it. Yeah. And there are wires, correct? These wires are actually tubes which are connecting, and here we can circulate the media. So this is the hollow fiber, a hollow fiber methodology for treatment and cultivation of an organism. And here the one which you see it on A is a most simplified way. Yeah, it is just an micro droplet, or even I'll explain you more simple way. Can you see some filter papers here?
Yes, sir.
Yeah. So in one of the filter paper, you put a soil, and then you put another filter paper here, and this kind of metal disc which you see, you put your media here. What happens is there is a diffusion of an organism will take place, and they will come into this media, and they will grow, and they will look like this. Can you see? These are some of the growth which we can see. So these are the two different chambers, and a methodology is an important step in the methodology for cultivation. I'm sure today definitely you all will find this very confusing, this hollow fiber chamber and diffusion chamber. Yeah. So just what you can do is today you all go back, or right now also I'll give you 10 minutes to read it. So just read this. If you can just come down onto, I know you all are on your phone, so it might be very difficult for you to see diffusion chamber, read it from here. So once you have a time, open it onto, or the notes which I've sent it, you can take a printout and then start reading. And these two things you read today, diffusion chamber and an isolation chip, and hollow fiber membrane chambers. And you come tomorrow, I mean Wednesday when we will first discuss this point, and then we will move further into this. Okay?
Yes, sir.
Yes, sir.
Yeah.
Yes,
So
sir.
what? Just to summarize today, what we did, on this, you can see this picture. First, environmental sample. So we talked about. See, if you see on your extreme right, there is a deep diver. Then there is volcanoes and some deep extreme environment, different extreme environment. The soil is being collected, and we are talking about culture dependent or traditional method. So we will be definitely designing a media, and then we are going to cultivate isolation and screening. So in isolation and screening, we were talking today about an isolation part. In isolation of an first soil collected, soils were treated as per the requirement and from the collection environment, and then we designed a media. With example I gave you for fungi, what kind of media, for bacteria, different kind of media, and then you can have your different kind of media. As we'll progress in your this different topics, we will keep on talking about the novel kind of media. So right now I'm just taking this two examples. So we talked about two media, and we put this soil, we treated this soil, and we try to put this onto the media for cultivation. So isolation, we are not talking about screening part. Isolation part we are still talking, and in that you need to describe two very important methodology. One is hollow fiber chamber and another is a diffusion chamber, which on Wednesday you will read, and I think you will put me a question and I will be trying to answer on hollow fiber and this diffusion chamber. Then we will move next towards a screening. with 3 examples. So the next class, what I'm planning is diffusion chamber and our hollow fiber model, followed by screening. And screening, what does screening mean? That we will discuss on the next class. Okay. Any questions or anything you want to discuss? I'm still here, but I'm otherwise closing the class here.
No, sir, no questions.
Okay, so this one PDF which I sent and the notes which I sent, don't discard, because there is short question. Your 10 to 15 mark question will definitely be from this aspect of methodology, because methodology is a basis and a fundamental to bioprospecting. Yeah, so we are right now talking about a classical methodology. Okay, thank you. Then I'll close the class. Yeah.
Thank you, sir. Thank you, sir.